and I promise I would stay within my time, or you can leave. So it was great to get Tony to present in ahead of me, because he actually gave half of my talk already, so should work. So we're a public company, and I'm going to make plenty of forward-looking statements, so don't trust everything that I would say, and read the information if you're interested in taking a position in TiGenix. Let me tell you, I'm going to do a completely different presentation to the ones I usually give, and the reason is, Tony has already explained everything you need to know about Chondrocelect and the cartilage product that we have, and they are doing the clinical trials against microfracture. We believe that the Chondrocelect has become today the real gold standard in cartilage regeneration, and I will have one slide there. And then we have 3 products on this adipose-derived stem cell platform, allogeneic use for inflammation and autoimmune disorders. And I will spend most of the time today talking about our rheumatoid arthritis 611 data, and that data is going to be presented at one of the plenary sessions at the ICR, which is the American College of Rheumatology Congress, one of the 18 papers only that is going to go through a plenary session this year on the 29th of October, so you're going to get the benefit of getting the data first. First on Chondrocelect, and again, I was on the panel and said that, it's tough to get reimbursement in Europe, but we're making some good progress. We got reimbursement in Belgium, being a Belgian company. We got reimbursement in the Netherlands, having a manufacturing facility in the Netherlands, and we got reimbursement in Spain, having half of the company coming from Spain. So if this is not politics, explain to me how it works. Unfortunately, we had nothing in France, so we got a negative decision in France, and we are partially reimbursed in the UK, partially reimbursed in Germany. And with that, and that is a 2.5 years process since we got approval, we've seen, and the numbers are very low, but we see the uptake that actually happens in Europe. So we got about close to 4 million sales last year. We're growing at 55%, and now we're getting, Spain and the UK, which are, 3 times each of those the size of the markets in which we're selling today. So we think that this will be a relatively small product. This will be, peak year sales are estimated to be at about 20 million euros, so it's a small compound with a relatively low margin. I think that the beauty is that this shows that, we have an organization that is capable of making a product all the way into the market, including manufacturing and getting the product reimbursed and commercialized. But then we believe that the real future of TiGenix comes out of this allogeneic adipose-derived stem cell platform, and the 3 products that we are developing, local and intravenous and intralymphatic administration of the cells for different diseases. Only one slide on 601. This is the, sorry, before we move into 601, why do we use these cells, and why do we believe that these cells are the right way to go? Out of the different mesenchymal stem cells, we still believe that adipose represents one of the best sources. They are very easy to access, you have a higher yield, and they are more stable and easier to grow. Also, we've demonstrated as well that they have a proven anti-immunomodulatory activity, and you have there the different mechanism, and something that is very important is we have now all the animal data that shows that they are very safe, no tumorigenicity, no toxicity, and actually also that they accumulate at the site of inflammation, which is where we need them to be. So I was going to say one slide on 601, which is our local delivery cells. And as you know, this is the most advanced of our clinical programs. We received orphan designation by the EMA, first ever cell therapy product to be granted orphan designation in Europe. It's currently in the middle of a phase 3 that will be a single pivotal phase 3. So if positive, will allow us to file for approval. We've been 7 times in front of the EMA to ensure that, from a regulatory standpoint, every development that we're doing will be agreeable by the EMA, and that indeed the product could be approved. So our preclinical program has been agreed. The CMC package has been pre-agreed by EMA, and the design of this single pivotal trial as well. It will be a very big trial, 278 patients that need to be recruited. We're in the middle of that recruitment, and we're scheduled to get the data by the end of 2014. And as I said, if positive, that will allow us to file in the first half of 2015. We have worldwide rights available. The idea is that we will commercialize the product ourselves in Europe, and we're looking for partners for the US and Southeast Asia. But again, I've presented a lot of times on 601 and the data and, the fistulas and the pictures. So let me move now into trying to get the presentation on 611 and our IV injection for rheumatoid arthritis. And, it's not only the clinical trials. Let me start by saying that, this clinical trial that we've just finalized came after a lot of work in animals, and this is on the chicken-induced arthritis model in mice, in which we inject, control or untreated mice versus the animals that were given, the ASCs concomitantly to the chicken collagen. And you see the dramatic effect of the cells on reducing inflammation. We've also compared then the cells to what happens if you compare them with either methotrexate or the anti-TNFs. And in this case, the results were not as nice, but they were comparable. So for the cells compared with either methotrexate or the today used anti-TNFs, the cell seems to have the same effect on this model. But that was in animals. Then we move into humans, and what we've seen is that when we look at RA patients, so we've done all these experiments in actually cells that come from our RA patients, that blood samples of RA patients, is that one is that the expanded adipose-derived stem cells in this patient population were capable of induced IL-10 secretion, which is important, and very importantly is that we are capable of reducing the pro-inflammatory cytokines that are created in those patients when you co-culture the PBMCs of those patients again with collagen II. So it seems to work in these patients. What happened locally? Well, we've done as well experiments looking at co-culturing the fibroblasts that are taken from the synoviocytes that are taken from the knees of the RA patients, and co-culturing them with human adipose-derived stem cells. And again, what we can see out of those experiments is that we can reduce the inflammation, and very importantly is the reduction in both collagenase and gelatinase, which are the two enzymes that are actually responsible for the damage that is caused in the cartilage on these RA patients. So again, we've moved from animals to the cells of the patients, and then, after all these experiments, we needed to see, but do they really work in real RA patients? And we did the first randomized clinical trial in this indication, and again, this trial was the first time that we were administering these expanded adipose-derived stem cells in humans, so it was really a safety trial. But it was, a relatively large trial of 611 plus a DMARD compared to placebo plus a DMARD. This is the standard design of these trials. 53 patients treated in 3 cohorts. The majority of those patients, 46, actually received the cells in 3 different doses. Very tough patient population, and we were expecting a tough patient population. You're going to see that we ended up with a very, very tough patient population because of the safety concerns at the beginning, they forced us to go to patients that have failed already 2 biologics prior to actually coming into the trial. And we were giving those patients just 3 injections, days 1, 8, and 15, and then following them without any further treatment of the cells for 6 months. We wanted to see, that was our hypothesis, that you will see a short-term effect, but actually the mechanism of action of the cells will prevent the patients from actually suffering some RA, the disease, for a longer period of time. So we wanted to follow those patients without any further treatment for 6 months and see the long-term effect of the cells. And primary endpoint was safety, but we follow every single endpoint that you can imagine on a clinical RA trial. So all the ICR criteria, but EULAR, imaging, quality of life, what have you. And I said that this was a very, very difficult patient population. And the average patients that we got, to our surprise, was a patient that had been on treatment for 13 years without success. On average, the DAS28, which is the disease activity that is measured on those patients with the C-reactive protein, was higher than 5.1, which is considered to be a very severe disease for all the patients in all the groups. The patients, on average, have received 3.3 non-biologic DMARDs prior to getting into the trial, with 75% of the patients actually receiving 3 different non-biological DMARDs. But on top of that, they receive, on average, 3 biologics. And again, 45% of the patients receiving 3 or more biologics. And again, people think of, well, which biologics did they get? Well, you have it here. So on top of the, the typical ones, Enbrel, Humira, and Remicade, for the majority, again, of the patients, most of the patients were rescued, even with Rituxan or Actemra. So basically, those patients, after 30 years, they've been through every single thing that doctors could throw at them without success. So that was, unfortunately, the patient population in which we had to show success with just giving them 3 injections and nothing else for 6 months. First, safety, because this was a safety trial. So the first thing that we can say is that we were very happy with the adverse events profile of the cells. Those, again, are very severe patients. Side effects of the biologic treatments that we're going to be compared with are extremely severe. Again, Humira, being the best-selling drug today in the world, has a black box warning with 5% of tuberculosis. So that gives you an idea of how severe some of the side effects associated to biologics. Out of 46 patients, we only got 1 patient that discontinued due to adverse events. That was on the lowest dose, actually. But all the other side effects were actually very mild. All were mild, actually, with fever and headache being the most common one. So nothing really that was worrying. And no signs of hematological toxicity, which is what one would expect with these treatments. So with that safety side effects, we went into the efficacy. And again, remember, those are patients that have not responded to anything. So I've just chosen 2 of the variables just to try to give you an idea of the indication of efficacy of the cells in this very difficult-to-treat patient population. And you have here, the red bar is placebo. We need to be very careful in explaining the data. There's only 7 placebo patients, so each patient will make a big jump. But it's normal, I think, to find in this very difficult-to-treat patient population that you get a very high placebo effect at the beginning as patients want to feel better after 13 years of failure, but the placebo effect really wears off, and this should be an M3. So by month number 3, none of the placebo patients, of course, is getting any benefit. And what is pretty remarkable is, despite the fact, remember, we've given them 3 doses before even finalizing month 1, at the end of month 3, still 40% of the patients, and this 46 is the number, so it's a pretty large number of patients, were moderate responders. And actually, 6 months after being treated with the cells, still 25% of those patients were being moderate responders. More importantly, if you look at DAS28, which is remission, how many patients were cured of this RA, 9%, but let me tell you, that means 4 patients. 4 patients were completely symptom-free 6 months after receiving just 3 injections of the cells. So again, very low numbers, but we believe, impressive results in this very, very difficult-to-treat patient population. So that's the data that will be presented at the end of the month, and that is the data that we're working now with the top opinion leaders of the world to try to define what should be the next step to move 611 into a phase 2B in RA. Thank you.